Glucose is the most important source of energy in many organisms. The regulation of glucose is vital to having a functioning body. In this video, we will explore the concepts of the target range for blood glucose levels, the function of the pancreas in glucose regulation, and the role and mechanisms of insulin and glucagon at a cellular level. Glucose is an integral component that plays a key role in metabolism. Glucose is a monosaccharide, or simple sugar, that is introduced into the body from carbohydrates, fats, and proteins. A normal level of glucose in the bloodstream is necessary to function. The target range for glucose in the blood is between 70 and 120 milligrams of glucose per deciliter of blood. An amount higher or lower than this range can be dangerous to the body if maintained for an extended period of time. An abundance of glucose may lead to a state known as hyperglycemia. Constant elevated glucose levels may lead to eye, nerve, and kidney disease, and in serious cases, diabetes. On the other extreme, a low amount of blood glucose, known as hypoglycemia, may lead to exhaustion, lethargy, and potentially a coma or death. A healthy body is able to regulate this delicate balance through the creation of hormones. The main hormones involved in glucose regulation are insulin and glucagon. Insulin and glucagon are antagonists. They work against each other to balance glucose levels in the bloodstream. When glucose levels are increasing or decreasing, a signal is sent to the pancreas to trigger the release of the appropriate hormone. Insulin will prevent the blood glucose levels from getting too high, while glucagon will prevent levels from getting too low. This negative feedback loop is an essential key to maintaining homeostasis in the body. Let's explore a bit more about where these hormones come from and what mechanisms they use. The pancreas is an organ that plays many roles in digestion. One of the key functions of the pancreas is to produce the hormones insulin and glucagon. Inside the pancreas are what are known as the islets of Langerhans. The islets are composed of a series of cells that release these hormones. Insulin is produced from the beta cells, and glucagon is produced from the alpha cells. Let's take a closer look. As a result from eating sugars or carbohydrates, glucose levels rise in the body. The beta cells of the pancreas are triggered and release insulin into the bloodstream. Insulin plays a key component in assisting glucose to pass through cell walls. A cell membrane serves as a barrier and is comprised of a lipid bilayer along with various protein receptors and channels. Glucose may not penetrate the cell membrane on its own. The role of insulin is to activate the glucose transporters that cross the cell membrane. This is known as facilitated diffusion. Two insulin molecules will bind to a receptor. That sends a signal to the glucose transporter to open. One glucose will bind to the transporter protein and will be moved through the plasma membrane. Once inside the cell, glucose may undergo several different processes to create or store energy. Primarily, glucose is converted into energy the body can immediately use. The energy is known as ATP, or adenosine triphosphate. This step is irreversible, as the ATP is consumed by the cell. A second process involves storing glucose as a larger molecule. Glycogen. Glycogen is a short-term storage of energy kept in muscles and the liver. This is a reversible process. Finally, glucose can be stored as lipids or fatty acids. The formation of adipose tissue, or fat, serves as a long-term storage of energy. This is an irreversible process, meaning the fat cannot be converted directly back into glucose. Together, these three processes prevent the body from entering a hyperglycemic stage as a result of having high glucose blood levels. Once these processes are underway, the amount of glucose in the blood will decrease, signaling the pancreas to begin the release of glucagon and lower the amount of insulin being released. This will typically occur in the time between meals, when sugar is not being introduced to the body and insulin has lowered the amount of blood glucose. Glucagon's role is to increase blood glucose levels and interacts with the cell in a different mechanism from insulin. Glucagon will bind to a receptor protein on the outside of a cell membrane and activate a signal inside the cell wall. This signal notifies the cell that there is a deficiency of glucose and triggers 3 different processes to begin. The first process caused by glucagon activation is the breakdown of glycogen, or the short-term storage mentioned earlier. This will release glucose back into the cell, ready for consumption. The second effect of glucagon is a process that results in the synthesis of glucose from non-sugar sources. Glucose can be created from lactate, formed by skeletal muscle during exercise, amino acids from proteins in food, and glycerol from fatty acids. The final process only occurs in a period of starvation. This is known as ketogenesis, or the creation of ketones. Glucagon triggers an irreversible process that breaks down fatty acids and certain amino acids into ketone bodies. Ketone bodies assume the role of glucose and provide energy that can only be used by the heart and brain. This direct source of energy to the brain and heart ensures that your body will continue to function when access to food is extremely limited. These main functions of glucagon, the breakdown of glycogen, the synthesis of glucose, and ketogenesis, work in tandem to provide energy to the body until food is reintroduced and the cycle restarts. As long as the body remains healthy, these processes will work in unison indefinitely.